Yeah, good day, everybody. We are 5 minutes past noon, and thank you, Kristin, for this very nice introduction, and the BFR, Tanja Schwertle, for inviting me to give that presentation to give you a view on the genotoxic compound risk assessment for pharmaceuticals. First of all, a short disclaimer. I'm not representing any EU competent authorities here, and a quick overview over the presentation. I'm just have 2 slides on the current approach for pharmaceuticals. Then I'm going over some challenges with this current approach for risk assessment and for regulation of pharmaceuticals. Potential advantages we may have from quantitative genotoxicity and the current challenges which are preventing currently the use of quantitative genotoxicity for risk assessment. Requirements to overcome from my personal opinion for the current limitations and some conclusions. And the current approach is laid down for active pharmaceutical ingredients by ICH S2R1 and S9. This is the guideline for advanced cancer treatment. And here we have genotoxic potential of the API may be acceptable based on risk assessment, and that holds true for compounds which have a genotoxic potential, but this is triggered by indirect mechanisms of genotoxicity where we can just derive or where we can fix a safe level, a threshold, due to the genotoxicity assays. Some examples there are fluoroquinolones or NRTIs, which are acting by indirect mechanisms and not are directly reacting with the DNA. And there, usually a safe dose is derived by using in vivo genotoxicity assays, deriving there the no observed genotoxicity effect level using safety margins to ensure that the doses the patients are treated with are safe with respect to genotoxicity, so the patient should not endure any genotoxic risk from the compound. And then we have usually the compounds where genotoxicity is the active principle. So all the alkylating anti-cancer treatments, for instance, are those examples, or others like the cyclins, and there the risk and the acceptability of the compound for treating the patients is due to a benefit-risk evaluation where the therapeutic benefit needs to outweigh the risk. So cytotoxic anti-cancer drugs are the best example for that. And then we have a different approach, slightly different approach for the impurities in pharmaceuticals, so compounds which are not contributing to the treatment effect. And they contain genotoxicants that are also not DNA reactive, and the first assessment there is basically a QSAR approach, which predicts whether the compound is positive in the Ames assay. And if that is negative, then we're usually regulating it up to 1 milligram per day of exposure as acceptable. Above that, or the 0.15% of impurity content in the API is the trigger for doing more assessment, but it's basically not considered as a mutagenic compound. And for these compounds, usually then also we can derive a permissible daily exposure level based on no observed genotoxicity effect level if the data are present. The focus of this guideline, and for us as regulators in the pharmaceutical area, is the focus on the DNA reactive substances. So at least this would be something like alkylators, which are directly causing DNA damage. We have heard a lot in the introductory talks this morning from especially Paul White about all the thinkings and all the data which are coming up now, also showing that such compounds may have practical thresholds. This is also mentioned in the M7 guideline. However, the problem is that for just showing that there may be a practical threshold for a specific compound, there is very, very challenging setting limits here based on a weight of evidence approach, including sufficient in vivo mutation data, mechanistic data, and so may there be possible even for these compounds on a case-by-case basis. However, I have not seen such an assessment yet. So basically, these compounds are therefore then not be regulated on that with such a risk assessment currently. So in vivo mutation data, that's also clear in the guidance and in the new Q&A, which has been published in July, I think, this year, that in vivo mutation data alone are currently not sufficient for deriving a safe level for these DNA-reactive compounds. And therefore, these DNA-reactive compounds are regulated by applying acceptable intake levels, and these are calculated by a linear back extrapolation from cancer data, from most of the cancer data in the CPDB, and there the back extrapolation for a theoretical 1 in 100,000 additional cancer risk leads then to the leads then to the TTC, the threshold of toxicological concern, which you already have heard about in the previous talks. And the TTC for contaminants in pharmaceuticals is 1.5 microgram per day. That's derived from the CPTB data, and that excludes, however, the compounds in the cohort of concern. That's aflatoxin-like, anitrosol-like compounds, and others, where we have a high concern that the carcinogenic potency is much higher and the linear back extrapolation for a 1 in 1,000 risk would lead to a threshold which is far below 1.5 microgram. These are usually then regulated by the most sensitive or most potent compound in that class, or by a class-specific TTC, which is derived then from the compound class carcinogenicity data, if there are enough there. That's basically also some of the problems there. So it needs the conduct of new carcinogenicity studies for contaminants, and that's basically usually not possible. We cannot do additional carcinogenicity data just to regulate contaminants. And therefore, the problem with, and Tanya has also said that in the introductory talk, the analytical capabilities are increasing, so we're identifying more and more impurities also in pharmaceuticals, and as these are also chemical processes, we frequently have then DNA-reactive mutagens identified in APIs and finished products. It's very often very challenging or even impossible to limit such impurities below a TTC, and for even the cohort of concern compounds with class-specific TTCs, even in a nanogram per day range, that's really a big challenge. It also leads, because also in this cohort of concern compounds, nitroso compounds are a very big example at the moment there, the mutagenic potency of these compounds is not the same for all the compounds. There are compounds with a high mutagenic potency and compounds which probably have very low mutagenic potency. And there, we probably have the problem that we are, for lots of that compounds, over-conservative with setting these really low nanogram limits there. So this, in the end, ends then up with the risk that for some investigational products, the development ends with identifying such contaminants in the product itself. And that may also lead that we are losing a lot of therapeutic compounds which may be good for treatment of patients but are not going further in the development because of the identification of these contaminants in there. And then I'm at the potential benefits of validated quantitative genotoxicity data. It would help us to get rid of the dependence on carcinogenicity data sets for doing the risk assessment, because we cannot do any more carcinogenicity studies. And in cases where it would be possible to conduct an in vivo gene mutation study to provide sufficient data for quantitative analysis and risk assessment of mutagens, health protective limits can be defined on the mutagenic compounds. Problem here is also, at least for the pharmaceutical area, we are talking about contaminants and impurities in there, and it's often very, very challenging to even get enough compound to even do a study there. However, such data would really increase the databases and inform the development of more sophisticated in vitro to in vivo extrapolation models. So we need this in vivo mutation data also to drive forward new models, as Paul has introduced a lot of that this morning already. It would also be helpful for informed developments to improve QSAR models. As you heard, the first assessment for mutagenic impurities is a QSAR model predicting the Ames test. And to help further developing those models to really calculate the mutagenic or even carcinogenic potency of those compounds would be also informed by such quantitative analysis of in vivo gene tox data. It would also inform models to quantitatively correlate mutagenic potential with carcinogenic potential of mutagens. That would be a short-term goal from my perspective, because a lot of data are already there. They need to be analyzed, and there it needs to be proven or shown that there is a correlation, that we can make a correlation between the mutagenic potency and the carcinogenic potency for these mutagens. And that would probably also lead to models to predict carcinogenic risk levels using quantitative genotoxicity data. Challenges with that: currently we don't have acceptable exposure limits that can be calculated based on currently we have, or we are only doing the calculation of limits based on theoretical cancer risk, which is theoretical because it's a very wide extrapolation and theoretical extrapolation. And it's known that the increase of mutation increases the risk of cancer. However, any mathematical correlation between that, can we just correlate the mutagenic potency with the carcinogenic potency? That's currently unknown. The data probably, or data sets are there, but the analysis should be done as soon as possible. And calculating a theoretical numerical increase in the cancer risk based on genetic toxicity data is currently not possible. And what do we need to overcome that? Evaluate, as I already have said, the in vivo mutation data and carcinogenicity data of the same compounds to just look whether the mutagenic potency and the carcinogenic potency correlate. And it then needs to be done to develop and validate methods establishing an AI or PDE. Paul has introduced a nice model there, and also all the work behind that, based on and developed that based on in vivo mutation data. And therefore that we need to define the most reliable and sensitive methods for measuring the endpoint. So TGR is one, error-corrected next-generation sequencing, duplex sequencing, all these techniques there obviously show, or the data which are out there, that they are even in sensitivity or even better in sensitivity than the TGR, and would have the advantage in future that we don't need transgenic animals for that. And we need to define the point of departure to be used for calculating that, and define the uncertainty factors to be included in the calculation of limit. Are the ones we have enough? Do we need more? Paul has also talked about that this morning. We agree internationally, that's very important in the pharmaceutical field, upon the methodology to calculate AIs or PDEs based on in vivo mutation data. And in a far future setting, because of we are all the time challenged with the 3 Rs, reducing, refine, replace, that all of these data are needed to inform and overcome the challenges for quantitative interpretation with in vitro with in vitro data, and there it's very important that we use the data to develop reliable and validated in vitro to in vivo extrapolation models. That's, from my perspective, very necessary here. And in conclusion, quantitative genetic toxicity is used currently for not DNA-reactive compounds, impurities, and active ingredients in pharmaceuticals. The use of quantitative genetic toxicity data evaluation for risk assessment of mutagenic compounds currently is not sufficiently validated for deriving acceptable exposure limits, and we need to overcome that by validating quantitative interpretation of gene tox data and quantitatively correlate mutagenicity potency with carcinogenicity potency. That would be the first step to go even far beyond overusing carcinogenicity data anymore, and just can predict even the cancer risk or lack of cancer risk based on mutation data and quantitative interpretation. Thank you.